Development Pharmaceuticals in association with Higher Secondary Principals Forum.
Hello students, we meet again for the biology class. We discussed in the previous class sexual reproduction in flowering plants, wherein we spoke about the structure of the stamen, the microsporangium and the pollen grain. We also saw microsporogenesis, that is the formation of microspores. Later on we moved on to understand about the carpel, the megasporangium and the embryo sac structure, wherein we discussed the 7-celled 8-nucleated condition of the embryo sac, which is the characteristic feature of the angiosperms. So today we will be discussing about pollination, double fertilization, outbreeding devices, pollen pistil interaction and post-fertilization events. Before I move on to pollination, let us try to understand what we had done in the previous class. It is just a revision for you. We saw the structure of the ovule, the Anatropous ovule, where I told you the Anatropous ovule has the stalk called as a funiculus and the body. The body is comprised of the outer integument and the inner integument. The body has 2 poles. This pole is the chalazal end or the chalaza, and this is the micropylar end or the micropyle region. Micropyle is opening. As you can see, this is the opening to enter inside the body of the ovule. On the inside of the body of the ovule, we saw that there is a mass of cells grouped together. This is the nucellus. Nucellus tissue is made up of the nucellar cells. And one of the nucellar cell later on transforms to behave as the megaspore mother cell, which undergoes meiosis to become the embryo sac. So this is what you see inside the ovary, the Anatropous ovule placed inside the ovary, and the Anatropous ovule is connected to the inner wall of the ovary with the help of the funiculus, which is attached to the placental tissue of the ovary wall. We come to pollination today. Now what is pollination? Pollination is the transfer of pollen from the anther to the stigma of a flower. Now when do pollens get transferred? Pollen grains get transferred once they are matured. And when are they matured? Either they are when they are in their 2-celled condition or in their 3-celled condition. So what happens when the pollen grains get matured? They are going to be dispersed through the line of dehiscence. If you remember, in my previous class we discussed what is line of dehiscence, which is along the longitudinal side of the anther lobes. So the line of dehiscence is the region which is going to rupture longitudinally, releasing the matured pollen grains into the environment. And these pollen grains are then later on going to be transferred to the stigma of the flower with the help of pollinating agents if it is cross-pollination. So let us try to understand the types of pollination here. We have autogamy, geitonogamy and xenogamy. Now what is autogamy? Autogamy is self-marriage. In simple words, it is self-marriage. So what is autogamy? Self-marriage. It is a transfer of pollen from the anther to the stigma of the same flower. So it is also known as what? Self-marriage. What is autogamy? It is the transfer, here, self-pollination it is also known as. It is a transfer of the anthers, of the pollen grain from the anthers to the stigma of the same flower. So this becomes self-marriage, also known as self-pollination, also called as your autogamy. Now, autogamy is commonly seen in a lot of flowers, but let me focus on a plant which is called as Camelina plant, which bears two types of flowers. Besides Camelina plant, you have the other plants like Viola, and yes, Viola is another type of a plant which is going to be showing you two types of flowers. Now what are these two types of flowers? As you can see over here, this is the same plant, the Camelina plant, but it bears two types of flowers. One is the open flowers called as a chasmogamous flowers, and the other ones are called as your clistogamous flowers or your closed flowers. This is the magnified view of the closed flowers which are in their bud condition. This is the clistogamous flower or the closed flower which is in its bud condition. So why are we talking about Camelina plant here? Because Camelina plant shows you here. This is the open flower or the chasmogamous flower, which is a bisexual flower. So it can show you what? Autogamy. Now, closed flowers, also known as clistogamous flowers, will only show you autogamy, and it will never be able to show you any other type of pollination. Isn't it so? Why? Because they are closed, they never open. The petals will not open to expose the anthers and the carpels. So here, it is definitely going to be autogamy or self-pollination. But the open flowers, though they are bisexual, they can show self-pollination or they can also show cross-pollination. So Camelina plant bears two types of flowers, open flowers called chasmogamous flowers and closed flowers which are known as your clistogamous flowers. Next type of pollination is geitonogamy. It is called neighbor marriage. Geitonogamy, also known as your neighbor marriage. Now why is it called neighbor marriage? Look here. If you notice, this is one plant. It has 4 flowers on it, right? Okay. Now what will be geitonogamy? It will be the transfer of pollen from the anthers of one flower to the stigma of another flower of the same plant. So it becomes what marriage? Neighbor marriage. Transfer of pollen from the anther of one flower to the stigma of another flower on the same plant. So functionally this becomes cross-pollination because you would require pollinating agent to transfer the pollen grains from this flower to the other flower. But genetically this is still going to be what? Self-pollination or autogamy. So geitonogamy, if you notice, functionally it is going to be your cross-pollination, but genetically it is going to be your self-pollination. Finally, you have the third type, which is xenogamy. Now in xenogamy, it is called a strange marriage. Now why is it called strange marriage? Because you notice that the transfer of pollen grains will be from a flower of a different plant to the stigma of a flower of another plant. So there are 2 different plants involved here, and it results in the transfer of the pollen grain from one flower to the other flower, and mind you, there are 2 different plants involved. So this becomes your cross-pollination, and it is going to be achieved with the help of, again, pollinating agents. So xenogamy here is the only type of pollination which is going to be bringing about genetic variation because there are 2 different plants involved in this. Whereas autogamy and geitonogamy, what do you notice? It is a type of self-pollination where there is not going to be variation that is going to be noticed because it is a type of self-pollination. Let's move on. Now we'll discuss about the pollinating agents here. We have different modes of pollination. We have water pollination, also known as your hydrophily. What is water pollination? It is the mode of pollination or the transfer of pollen through the agency of water. So you can see over here, this is a plant, aquatic plant called as your Vallisneria. You remember in my previous chapter when we discussed about vegetative propagation, the vegetative propagules, one of the vegetative propagules was offset. So this is what an offset is, which is like a runner found in your aquatic plants only. Moving back to what we are discussing about pollination here. So pollination in water is also known as hydrophily through the agency of water. Examples of plants which carry out water pollination are Zostera, Vallisneria, Hydrilla and Lemna. The example that is put up over here is Vallisneria. As you can see, it is a plant which produces male flowers, and Vallisneria, the other one which produces female flowers here. Now you also notice something, that the male flowers will be released onto the surface of the water. And the female flowers are very long, that means they have a very long stalk, and their other parts of the flower are going to be exposed on the surface of the water. So where does pollination actually take place? Pollination takes place on the surface of the water body in Vallisneria. So what will happen is the pollen grains from the flowers, male flowers, which are on the surface, are going to be taken or transferred to the stigma of the female flowers or the pistillate flowers. And how are they going to be transferred? It could be with the help of water, that is wind pollination. That's why it's called hydrophily or water pollination. That is the agent involved here. But it also could be with the help of insects, students. Isn't it so? Male flowers are present on the water surface. Female flower is also there on the water surface. So even insects could be the agents involved to transfer the pollens from the male flowers to the female flowers. But what kind of pollination is this? Water or hydrophily. Now, what are the nature of the flowers in this case? In these plants, the flowers are going to be very small and inconspicuous. They are not going to be having any nectar or odor in them. As it is, they are very small. They are not going to be having their proper whorls also. The pollens are going to be light and unwettable. So also will be the stigma, which is going to be unwettable, long and sticky. Now what is the reason for the pollen grains to be unwettable, the stigma to be unwettable? Because see, if water enters into the pollen grains, what will happen? They will become heavy and they can sink. For that reason, the pollen, the stigma are unwettable. And how are they unwettable? Because they are coated with mucilage. Wind pollination is the next one. It's also known as anemophily. How do you define wind pollination? It is the mode of pollination or transfer of pollen through the agency of wind. Now examples: coconut, date palm, maize, grass, etc. So these are the plants that will carry out wind pollination. Now in this wind pollination, what should be the nature of the flowers? Wind pollinating plants, what should be the nature of the flowers? The flowers are colorless, odorless and nectarless. Do you require odor, color and nectar laden inside such flowers? No, because they are going to be transferred, the pollens are going to be transferred with the help of the wind or the air. So the pollen should be light, they have to be small, they have to be winged or dusty, dry or smooth and non-sticky. So these are the characteristic features of the pollen, because these features only will help the pollen to be transferred with the help of wind. What should be the nature of the stigma? The stigma in the female flowers should be hairy, feathery or branched, so that they are able to trap the pollen grains which get transferred with the wind. And the pollen are produced in large numbers. So you notice here there is wastage of pollen grain. Why? Because the pollinating agent involved is wind. Moving on to the next pollination, that is insect pollination, also known as entomophily. It is the mode of pollination or the transfer of pollen through the agency of insects. Now besides insects, are there other animals involved in pollination? Yes. So the transfer of pollination or the mode of pollination or the transfer of pollen through the agency of animals is going to be called as zoophily. And what would be the other animals besides the insects which are involved in pollination? They could be your mammals like your primates, example lemurs. We could also have bats and birds and some reptiles that can also bring about your pollination. So we'll discuss about your insect pollination now. In insect pollination, in insect type of pollination, what should be the nature of the flowers? The flowers should be usually large. They have to be scented. They have to be having nice, large, bright petals in order to, attract the insects towards them. They also have to be richly laden with nectar. It's like a reward for the insects to come to them. And the pollen, what should be the nature of the pollen? The pollens have to be sticky or spiky so that they can adhere to the body wall of the insects. And the pollen production is moderate. You don't require the pollen production to be in large number. And what should be the nature of the stigma? The stigma has to be sticky so that the pollens can, get attached to it. We have discussed about pollination and why is pollination required or needed, so that you can transfer the pollen from the anthers to the stigma. The moment the pollens reach the stigma, the pollen grains will germinate now. They will release the pollen tube through the germ pore, and the pollen tubes are now going to be carrying the 2 male gametes which are involved in fertilization. Before that, let us discuss about outbreeding devices. What are outbreeding devices, students? Outbreeding devices are mechanisms which are followed or adapted by plants to prevent self-pollination. Now, all this while we discussed 3 modes of pollination: autogamy, geitonogamy, xenogamy, and we also saw about the pollinating agents. But now I am trying to make you understand about certain devices or mechanisms that the plants have been using, and they are called as your outbreeding devices. So they are mechanisms that are used by plants to prevent self-pollination. So plants usually prefer cross-pollination and they would always try to avoid self-pollination. Why is it so? Why do plants prefer cross-pollination over self-pollination? Because continuous self-pollination in plants could lead to a phenomenon which is called as inbreeding depression. Now, what is inbreeding depression? Inbreeding depression is something where you notice that the plant loses it basically loses its vigor, its vitality, its productivity, and the genome of that particular plant gets weakened. So the offsprings that are going to be produced are going to be weak offsprings with weak genotypes. Okay? So to avoid that, the plants would always prefer to go for cross-pollination, because what does cross-pollination do? Cross-pollination is going to be helping these plants to revive back from inbreeding depression. What does cross-pollination do? Cross-pollination brings about genetic diversity. So how do plants bring about or promote cross-pollination and avoid self-pollination? These are the ways by which the plants will be doing it so. So let's see the first one. The pollen release and stigma receptivity is not synchronized in plants. Now, if it is a bisexual flower would try to basically carry out self-pollination. It doesn't have an option, right? Because if it is a bisexual flower, the anther will be there, and you are also going to be having the carpel, which is going to be carrying the ovary style and the stigma. Inside the ovary, you are going to be having the ovule. So if it is a bisexual flower, there are possibilities that this particular flower will be carrying out self-pollination. But how does this flower prevent self-pollination? So to prevent the self-pollination in such a bisexual flower, the pollen release and stigma receptivity is not synchronized, meaning what? The pollens get matured probably later than the carpels. Are you getting me what I'm trying to say? So if the maturity of the anther and the maturity of the carpel is in par, there are possibilities of self-pollination. Pollen release and the pollen get dusted onto the stigma, because stigma is also matured and ready now, and it would bring about self-pollination. But what if the pollens get released earlier than the maturation of the carpel? So if the carpel, the stigma is not matured, it's not ready, even if the pollens have already been released and transferred on the stigma of this particular flower, the pollen are not going to be germinating. Why? The stigma is not receptive, it is not ready, it is not matured. It could be the other way around also. It could be the carpel that gets matured earlier than the anther. So if the carpel gets matured earlier than the anther, what will happen? The carpel is receptive to pollens from outside and not the pollen of this particular flower. So it would prevent self-pollination and promote cross-pollination. Look at the next one, the different size and position of the stamen and the carpel. In a bisexual flower, how to avoid self-pollination and promote cross-pollination. Again, here you can go for the next one, which is different size or position of the stamen and the carpel, which means the stamen position could be and the carpel position could be in opposite direction. So if it is in the opposite direction, what would happen? It would always try to promote cross-pollination and avoid self-pollination because the position is different. The angles of placement of the stamen and the carpel is in different direction. Or it could be also the size. The size of the stamens and the size of the carpels matter. So if the size is different, then also it would promote cross-pollination and prevent self-pollination. The next one is self-incompatibility. Now this self-incompatibility, students, is something which you will see usually in bisexual flowers, where the pollen of the same flower is not going to be receptive, or the stigma is not going to be receptive to the pollen of the same flower. But the stigma is going to be receptive to the pollen of other flowers from different plants. So that becomes self-incompatibility. That means the stigma is not compatible with the pollen of the same flower, which is self-incompatible. And finally, we have unisexuality. If flowers are unisexual, or in other words, flowers are either male flowers or female flowers, you don't have any other option, but it is always going to be promoting cross-pollination rather than self-pollination. So unisexuality is a way by which you can always promote cross-pollination and avoid self-pollination. We come to the next topic, pollen-pistil interaction. Now what is this pollen-pistil interaction? This is a kind of a chemical dialogue that takes place at the level of the stigma, where the pollen have already come and they are resting on the surface of the stigma. So the pollens have already undergone pollination with the help of pollinating agents maybe, and the pollens have now come and they are seated on the surface of the stigma. And what are they going to be waiting now for? They are going to be waiting for the germination of the pollen tube, right? Yes. So here, do you think it is so simple that the pollen grains just sit on the stigma and the stigma is going to allow them to germinate? No, it doesn't happen so, because the stigma is very smart. The stigma is going to see whether the pollen grains are compatible with it. Only after it realizes the compatibility, then only it is going to allow the pollen grains to germinate. If not, if the pollen and the pistil is not compatible, the pollen grains will never germinate. And even if they germinate, the pollen tubes probably will get degenerated along the way of this style and they will never reach the egg. So as you can see over here, the moment the pollen grains come and lie on the stigma surface, there is a kind of a dialogue that takes place, a chemical dialogue between the stigma and the pollen grains. If you notice, the pollen grains are of the type S1 and S2. And what is the nature of the stigma and the style? It is also of the nature S1 and S2. That means these are the pollens from the anthers of the same flower where you have the stigma or the style that is located on. So do you think this particular stigma will promote self-pollination? No. It would try to avoid self-pollination. That means it will bring about self-incompatibility. As you can see over here, it's a big no to the pollen grains. The pollen grains are not going to be allowed to germinate. Now look over here. There are two pollen grains here, S1 type and the other one is S3. Now S3 is a type of a pollen which has come from another flower. And as a result, there is going to be an interaction between the pollen and the stigma. It's a chemical dialogue, as I told you. And with this interaction, you will notice the pollen tube now starts growing or germinating. It interacts with the receptors or the chemicals along the way in the stigma and the style, and it grows further. And what is it carrying along with it at the tip region over here? Please remember, students, it is carrying the two male gametes. The two male gametes are at the tip region of the pollen tube. So you can see over here, the pollens S3 and S4 have germinated. Why? Because they are of a different flower, from a different plant probably. Okay? And as a result, the stigma and the style are highly receptive to it, and they are compatible. With the help of the chemical dialogue, which is called pollen-pistil interaction, the pollen tubes now germinate, and the germinated pollen tube now will reach the ovule. Through the micropyle, it will enter inside the embryo sac. Did you see this? Okay. Now, how is it that the pollen tube grows and reaches the ovule through the micropyla to enter into the embryo sac? Again, it is the pollen-pistil interaction, the same chemical dialogue which is guiding the pollen tube, which is carrying the two male gametes to reach the ovule, the Anatropous ovule, through the micropyla region into the embryo sac. Now we come to double fertilization. Now what is double fertilization? Double fertilization is considered to be the significant characteristic feature found exclusively in angiosperms. One more characteristic feature that we have already seen is the 7-celled, 8-nucleated condition of the embryo sac in angiosperms, and this is another significant characteristic feature in angiosperms, which is double fertilization. So what happens in double fertilization? There are two male gametes. So one of the male gamete will fuse with the two polar nuclei in the central cell of the embryo sac to form the primary endosperm nucleus, and then the primary endosperm nucleus now becomes triploid, 3N. The central cell of the embryo sac had two polar nuclei, N and N. So the moment one of the male gamete reaches the polar nuclei, which is again N, so it is going to be the fusion of the three nuclei resulting in a triploid primary endosperm nucleus. And what happens to the other male gamete? The other male gamete fuses with the egg cell. Remember egg cell, the egg apparatus in the embryo sac? The egg cell which is guarded on either sides by the two synogyds. The egg cell is a haploid cell, N, and the other male gamete is also a haploid cell, N. Okay? So when N and N come together, the haploid nature, okay, of the gametes, when they fuse, when the gametes fuse, you are going to be getting the zygote and you are going to get the diploid nature restored back again. So the other male gamete fuses with the egg cell to form the zygote. Let's try to understand what happens. I want you to look at the figure over here, and I want you to understand how exactly double fertilization occurs. So what you notice here now, if you see this, this is the pollen tube which is carrying the two male gametes. In this figure, you can notice the pollen tube which is carrying the two male gametes enters the embryo sac through the micropyla region, and the pollen tube enters into one of the synogyds. That means the synogyd, one of the synogyd will help the pollen tube, it will guide the pollen tube to enter into the egg apparatus or the embryo sac. So the pollen tube carrying the two male gamete pierces through one of the synogyds, it breaks open and releases the two male gametes. So one of the male gamete, as you can see here, the two nuclei, the two male gametes have already moved out from the pollen tube. One male gamete goes and fuses with the egg cell. The other male gamete goes and fuses with the primary, in the central cell with the polar nuclei to form the endosperm, which is triploid, and this egg cell becomes now the zygote, which is diploid. This is your double fertilization. So in double fertilization, let's summarize what happens. You see one male gamete fuses with the egg cell to form the zygote, a diploid zygote. The other male gamete goes and fuses with the two polar nuclei and becomes a triploid nucleus, which is the primary endosperm nucleus, which later on will become the endosperm. In post-fertilization events, we'll discuss about endosperm formation and embryogenesis. Endosperm development. Now, how does the endosperm develop? what is endosperm in the first place? Endosperm is a nutritive tissue. And what type of a nutritive tissue is it? It is a triploid nutritive tissue. It is made up of cells which are triploid, 3N. From where does the endosperm tissue originate, students? It originates from the primary endosperm cell which carries the primary endosperm nucleus. We have just discussed about the primary endosperm nucleus. And where is the primary endosperm nucleus found, which is triploid? It is found in the central cell. But the moment fertilization occurs, when one of the male gamete fuses with the two polar nuclei in the central cell, the two polar nuclei and the male gamete, when they fuse together, it causes the formation of the primary endosperm nucleus, which is triploid, and the central cell is no longer going to be called as a central cell. It is going to be now called as the primary endosperm cell, carrying the primary endosperm nucleus. This primary endosperm nucleus now, here, you can see this. This is the primary endosperm nucleus. Inside this cell, this was earlier the central cell, but now it is the primary endosperm cell which is carrying the primary endosperm nucleus. This is what, students? This is your zygote, the diploid zygote. And what are these? These are your 3 antipodal cells at the opposite end of the embryo sac. This whole structure is your embryo sac or the female gametophyte inside the ovule. But now the terms have changed. The terminology is different after fertilization. Primary endosperm nucleus, primary endosperm cell, this is the zygote, and these are the degenerating antipodal cells. So what happens to the single primary endosperm nucleus here, which is triploid? It starts undergoing mitosis, mitotic division, resulting in only karyokinesis, not cytokinesis. So what happens is only the nuclei start dividing and not cytokinesis. That means cytokinesis doesn't take place as yet. So when only the nuclei are dividing, when 1 becomes 2, 2 becomes 4, 4 becomes 8, what you notice is only the division of the nucleus and not the cytoplasm. So what you notice here now is going to be only free nuclei that are formed through mitosis. Free nuclei that are been freely circulating in the primary endosperm cell. And how are these free nuclei formed? Through mitosis and only with karyokinesis. Finally, what happens is the free nuclei later, you will see at the periphery region, slowly they start getting their cell walls. That means the cell walls are going to be laid for the free nuclei, and later on you will notice that most of the free nuclei will be laden with their cell walls, and now they are going to be your typical cells. Now, I have made a point over here. Coconut water is your free nuclear endosperm, and the white kernel of the coconut is your cellular endosperm. Have you ever given it a thought, students? We all enjoy drinking tender coconut water. But have you ever thought, what is that tender coconut water that you drink? Have you any time analyzed it or thought about what is this tender coconut water that you always drink? That is nothing but your free nuclei of the endosperm. So this type of the nutritive tissue, the free nuclei one, is known as your nuclear type of endosperm. So remember, students, the nutritive water that you drink is actually the nutritive tissue for the developing embryo. And that is a type of an endosperm which is known as the nuclear type of endosperm. Okay? So next time when you drink tender coconut water, remember what are you basically going to be having? You are going to be having the nuclear endosperm of the embryo sac. Okay? Then what is this white kernel? The malai, as we talk about, of the coconut. So you have the 2 varieties that you usually get in the market. You ask for either the tender coconut which is with full water, and the other one with a little bit of malai, right? What is that malai? It is nothing but your cellular type of endosperm. The moment the free nuclei okay, they undergo cytokinesis. What will happen is the cell walls are going to be laid, and as a result that tissue now is going to be the white part of the tissue, which is going to be the cellular endosperm. So you have the nuclear type of endosperm and you have the cellular type of endosperm. And what is cellular type of endosperm? The primary endosperm nucleus will divide, and as a result you will notice after karyokinesis the cytokinesis is also going to be seen. Cell wall is going to be laid and you are going to be getting two cells. So from one cell it becomes two-celled endosperm tissue. Then 2 will become 4, 4 will become 8, 8 will become 16 and so on. So here this tissue, this nutritive tissue is going to be your cellular type of endosperm, and in this case it is nuclear type of endosperm tissue because you have only free nuclei in the beginning which are not laden with the cell walls. Later you will see that these free nuclei, they get their cell walls, transforming them into the cellular type of endosperm. Next we move on to embryo development. How does the embryo get formed? It gets formed from the fertilized egg cell, which becomes the zygote, the single celled zygote, and from this diploid single celled zygote you are going to be getting the embryo. Now look here students, this is your zygote, the diploid zygote. This diploid zygote now carrying the diploid nucleus undergoes mitosis. So the first mitotic division results in two cells. One cell is going to be the zygote cell and the other one is going to be the suspensor cell. So you have this cell as the suspensor cell and this cell is going to be your zygote cell, which will become the embryo later. What happens to the suspensor cell? It also undergoes mitosis to form a long suspensor, and the zygote cell also undergoes mitotic divisions further to become the globular embryo. Have a look at the shape of the embryo, it is globular. As the embryogenesis progresses through mitotic divisions, you notice the suspensor increases by the increase in the number of mitotic divisions of the suspensor cells, and the globular embryo now transforms and becomes heart-shaped embryo. From heart-shaped embryo, you get the horseshoe-shaped embryo, wherein now you notice the embryo axis is also going to be noticed here. You will be able to see the cotyledons. If it is a dicot embryo, you will have two cotyledons. If it is a monocot embryo, you will have a single cotyledon or the nutritive tissue. You will have the plumule and the radicle also that is going to be seen. Now, I want you to understand the role of the suspensor, students. What does the suspensor do? If this is your zygote and this zygote cell here starts undergoing divisions, you would have this structure over here. Understand? You would have this structure, the globular embryo. I place it over here inside the embryo sac. The suspensor is growing and it helps to push the embryo into the nutritive tissue or the endosperm. Where is the endosperm located, students? This is the place where the zygote is, which will become the embryo, and this is the area where the endosperm is getting formed or is located. Why do you need the suspensor? You need the suspensor to push the embryo into the nutritive tissue. The embryo cannot walk towards the nutritive tissue. The embryo is feeding. It is made up of cells which are voracious feeders. It's growing, so it requires nourishment. And because it requires nourishment, you have the endosperm tissue. But you require something that will push the embryo into the nutritive tissue or the nourishing tissue, and that is going to be done with the help of your suspensor. So the suspensor grows, and as it grows, it pushes the embryo, which is at the moment globular, it becomes heart-shaped and then it becomes the horseshoe-shaped. And how does it change? Because it is growing, and it is growing because it is getting nourishment from this region, which is already now the endosperm tissue. So we have the two types of embryos. This is a typical dicot embryo with the two cotyledons. the plumule and the radicle with the root cap, students. And here you have the embryo of a monocot plant, example grass. You have the scutellum. There is no word cotyledon here because in grass the single cotyledon is known as scutellum. What is it called? It is called as the scutellum. Besides the single cotyledon called scutellum, you will also have the regions which are going to be responsible to give rise to the body of the plant. That is the shoot apex, the plumule region, the radicle with the root cap. You also have additional sheaths which are not found in a dicot embryo. One is called coleorhiza, the other one is called coleoptile. What is coleoptile? It is a protective covering enclosing the plumule. And what is coleorhiza? It is a protective covering that covers the radicle because these are sensitive tissues. Let's now discuss about the terms which are now going to be changed. So after fertilization occurs and embryogenesis begins, you will also notice that there are changes that take place in the ovary and the ovule as well. So what happens as the embryogenesis is progressing inside the ovule, students? You will see that the ovary wall, okay, becomes the fruit wall. It is also known as the pericarp. What is the ovary wall going to get transformed now into? It gets transformed into the fruit wall, also known as the pericarp. The ovary now becomes the fruit. The integuments of the ovule, the outer and the inner integument, remember, they go on to become the seed coats of the seed, the outer two coats of the seed. So the outer integument becomes the testa or the outer seed coat. The inner integument becomes the tegmen, also known as the inner seed coat. And the ovule now gets transformed into the seed, which is carrying what? Which is carrying the developing embryo inside it. So we have the two seeds, the dicot and the monocot seed. You have here the dicot seed and the monocot seed, the maize seed and the bean seed. Example, dicot and monocot. So as you see over here, we have two cotyledons. That's why it is a dicot seed. It has two cotyledons. The radicle, it has the epicotyle and the hypocotyle region. In this section you notice there is endosperm. There is the cotyledon also. And of course you are going to be having the epicotyle and the hypocotyle regions seen in the corn seed. So I want you to understand one thing. Here you have two cotyledons. That's important in a dicot seed. And in this case you have only a single cotyledon, which is the monocot seed. And of course you have your embryo axis in both the cases, that is in both the seeds, which is your radicle and the plumule, which will grow. And what are these two? These are the two coats, the testa and the tegmen. These are the two coats of the seed, which have come from the integuments of the ovule. You can see the differences between the dicot and the monocot seed now. The dicot seed carries two cotyledons, the monocot seed carries or contains one cotyledon. What are cotyledons? Nourishing or nutritive tissues in the seed. In a dicot seed, the germination is epigeal, and in a monocot seed, the seed germination is hypogeal. What is epigeal and hypogeal? Epigeal germination is the germination of the plant above the soil surface, and hypogeal germination is going to be germination of the plant below the soil surface. So in epigeal germination, you will see the cotyledons which are going to be above the soil surface, and in hypogeal germination, you will notice that the cotyledons are going to be below the soil surface. Examples of dicot seeds are pea, gram, beans, castor, papaya, and in monocot seeds we have your cereals and your grass plants. Now, in matured seed, students, you can categorize matured seeds into albuminous type and non-albuminous types. Albuminous types of seeds are also known as endospermic seeds. Non-albuminous seeds are known as non-endospermic seeds. It's very easy to understand. What do you mean by endospermic seeds and non-endospermic? Endospermic seeds are seeds which are going to have the endosperm persistent in them besides the cotyledons. So cotyledons are nutritive tissue, we all know that. Endosperm is also considered as a nutritive tissue, and it was the primary nutritive tissue for the developing embryo. So the developing embryo was initially fed with the help of the endosperm and not the cotyledons. Cotyledons come later during the embryogenesis. But the initial stages, you will notice it is the endosperm which is the nourishing tissue. But in a matured seed, when you have the endosperm which is still persistent and is still providing nutrients to the seed along with the cotyledons, this becomes an albuminous seed or an endospermic seed. And the non-albuminous seeds or non-endospermic seeds would be the ones where the endosperm is very much reduced or totally used up, and therefore these seeds are now going to be basically depending on the cotyledons. For example, coconut, castor, maize, albuminous seeds, and you have pea, beans, mustard, non-albuminous seeds. Let's move on to the type of fruits now. There are three types of fruit, students: true fruit, false fruit, and parthenocarpic fruits. What are true fruits? True fruits are fruits which are formed from the ovary, and no other part is involved in the formation of the fruit. So it's the ovary that gives rise to the fruit, and such fruits are going to be known as your true fruits. For example, your mango, tomato, these are examples of true fruits where the ovary develops into the fruit without any non-carpellary part involved in it. So it's only the carpel that is involved in the transformation or the formation of the fruit. That's why such fruits are going to be known as your true fruits. But what are false fruits? They are also a type of fruits which are edible, students, but you can categorize them as your false fruits. Why? Because they are fruits that originate from parts other than the carpel. So if you have a fruit that originates from a part other than the carpel, then you will call it as your false fruit. And most common part that goes into the formation of a false fruit is the thalamus. What is the thalamus? If you know, students, the flower, the floral whorls, the calyx, corolla, androecium and gynoecium, they are positioned on a disc, and that disc or the base is known as the thalamus. So it is the thalamus in the case of fruits like apple that go on to become the fruit, and such fruits are going to be known as your false fruits. Besides the thalamus, there could be, parts of the plant like the perianth, of the flower like the perianth, involucre, etc., which also develop into the fruit. What are parthenocarpic fruits? Parthenocarpic fruits are fruits which are developed without fertilization. We have already discussed this in the previous chapter about parthenocarpic fruits, and I had also told you it is not the same as parthenogenesis, right? Okay, so what are parthenocarpic fruits? Fruits that are developed without the fertilization, and these fruits are always going to be your seedless fruits. For example, bananas. Coming to the last part in this chapter, apomixis and polyembryony. These are special types of reproduction that you have in plants. Apomixis is a type of a special asexual reproduction, and you have the other one, that is polyembryony. So let us try to see what is apomixis here. Apomixis is a phenomenon in which a plant produces seeds without fertilization. A plant produces seeds without fertilization. So how will that occur? When you see a plant producing seeds without fertilization, it clearly means that there has been no meiosis and fertilization. That also means, understand, the megaspore mother cell, which is a diploid cell, in a normal situation would undergo meiosis to become 4 megaspores, and out of the 4 megaspores, which are haploid now, 3 will degenerate and only 1 remains to further get transformed into the embryo sac. Yes? But in this case, in plants which are called apomictic plants, which undergo apomixis, the megaspore mother cell, the diploid megaspore mother cell, does not undergo meiosis. It transforms directly into the diploid embryo sac, and you will have directly seeds that are going to be formed without fertilization. So, and look at this point. The seed genotype is consistent with the female parent. Do you agree with this point or not? Now, the seed which is formed now, the seeds that are formed are going to be having the exact copy, or it's going to be the exact replica of the female parent. That means the female genotype is going to be the same as the seed genotype, or they're going to be the clones. This is found in meadow grasses, blackberries, dandelions, etc. And what is polyembryony? Polyembryony, as you can see over here, this is a mango seed where you have a single embryo inside it, and here you have also again another mango seed which is having 2 embryos inside it. So usually 1 seed, 1 embryo. But here you have more than 1 embryo inside the seed, and this condition is going to be your polyembryonic condition. And polyembryony is mainly seen in plants like your citrus plants, your mango, etc. So what is polyembryony? It is the occurrence of more than 1 embryo in a seed. So you have more than 1 embryo in a seed, that condition becomes polyembryony, like how you have twins. Embryos can arise in the same embryo sac or in different embryo sacs in the same ovule. So how do you explain polyembryony in plant, students? I told you polyembryony is like how you get twins that are formed. How are identical twins usually formed? They are formed from a single zygote. That single zygote splits into 2, and as a result you will get identical twins. It is a single zygote splitting into 2. In the same manner, what happens here is when the male gamete, one of the male gamete fertilizes the egg cell now becomes a diploid zygote in the plant, right? Now that diploid zygote goes on to become the embryo. It would become a single embryo. But if it is a case of polyembryony, that single celled diploid zygote, if it splits into 2, what will you get now? You will get 2 embryos inside the single ovule, and that is a case of polyembryony. Besides the cleavage of the zygote to become 2 different embryos, you can also see that it could be any of the cell of the embryo sac that can behave as an embryo. We all know it is the egg cell that would fertilize with the male gamete and behave as the zygote. But due to some changes, it could be mutations that occur, desirable or undesirable mutations, wherein you will notice that any one of the cells of the embryo sac could transform or behave as the embryo inside the single ovule. It could be, usually it is seen that it is the synergids which are on the adjacent side of the egg cell, or which are the neighbors of the egg cell, that go on to become the additional embryos, resulting in the condition known as your polyembryony. Rarely it is also found that some of the nucellar cells also, they enter inside the ovule, that is the embryo sac, they pierce through the embryo sac and they get transformed into the embryos, resulting in polyembryony. Okay. So it is commonly found in mangoes and citrus plants. This is what we have seen now in this chapter, sexual reproduction in flowering plants. Let us go through some of the questions in this chapter. These are all your board questions, students. Let us quickly go through them and see the type of questions usually asked. Abundant food reserve material enclosed in the mass of cells within the integument is dash. What would be your answer? Within the integument, tapetum, endosperm, nucellus, chalaza. Answer is your endosperm. Define geitonogamy. Draw a neat diagram of the false fruit of apple. How is autogamy different from xenogamy? You need to differentiate between the two types of pollination. The wall layer of microsporangium that nourishes the developing pollen grain is dash. Endothecium, middle layers, epidermis, tapetum, it is the tapetal layer. Draw a neat diagram of the TS of a matured anther, the anther which has already undergone dehiscence. What is apomixis? Sketch the diagrammatic representation of a mature embryo sac. Name the most resistant substance covering the pollen grains. The answer is sporopollenin. Draw a neat diagram showing the LS of a flower. Have a look at this question. In maize, the chromosomal number in its meiocytes is 20. Hence, the number of chromosomes in its endosperm and egg cells will be dash. You have 4 options given, 20 and 10, 30 and 10, 40 and 20, 60 and 20. Have a look here. It is given to you that in maize plant, the chromosomal number in its meiocyte. What is a meiocyte? Cells which undergoes meiosis is known as a meiocyte. In a meiocyte, the chromosomal number is 20. So what could be a meiocyte that is going to be giving rise to the endosperm and the egg cell? Megaspore mother cell. And what is megaspore mother cell? It is a diploid cell. Having how many chromosomes? 20 chromosomes it is said. Now the question is, the number of chromosomes in its endosperm and egg cell will be dash respectively. That means endosperm we all know is a triploid tissue, 3N. Meiocyte, megaspore mother cell is diploid 2N. So triploid would be 30 chromosomes in the endosperm, and egg cell is a haploid cell formed through meiosis. So it is going to be 10. Haploid, which is half the number of chromosomes of the meiocyte, which is found to be 20. So half of 20 is going to be 10. So the answer is going to be the endosperm chromosome number will be 30, and egg cell the chromosome number is going to be 10. And these are the list of diagram, students, that you need to practice. Figure 2.1 from your textbooks, these are 2.3c, 2.7d, 2.8c and 2.15b, which is the diagram of the false fruit, that is apple. So we come to the end of the chapter, the second chapter in this unit of reproduction. I hope you have understood the chapter, students, and I hope that you will go through and practice the diagrams and solve the questions from various question papers. Thank you, dear students, and have a nice day.
Prudent Scholars, powered by Lupin Pharmaceuticals.